Performing a meta-analysis in medical research involves systematically reviewing and synthesizing existing research studies to derive quantitative conclusions about a particular research question or hypothesis. Here's a structured guide on how to conduct a meta-analysis in medical research. Understanding meta-analysis in medical research. Meta-analysis is a statistical technique that combines results from multiple independent studies on a specific topic to provide a more robust and precise estimate of effect size or outcome than individual studies alone. It enables researchers to identify patterns, inconsistencies, and trends across studies. Steps to perform a meta-analysis. Formulate research question and define inclusion criteria. Research question. Clearly define the research question or hypothesis that the meta-analysis aims to address. Specify the population, intervention, exposure, comparator, outcomes, PICO framework, and study designs of interest. Inclusion criteria. Establish explicit criteria for including studies based on relevance, study design, e.g., randomized controlled trials, cohort studies, and quality, e.g., risk of bias assessment. Systematic literature search and study selection. Literature search. Conduct a comprehensive and systematic literature search across relevant databases, e.g., PubMed, Cochrane Library, using predefined search terms and filters. Document search strategy and results. Study selection. Screen retrieved studies based on inclusion criteria. Assess eligibility through title and abstract screening, followed by full-text review of potentially eligible studies. Document reasons for exclusion. Data extraction and quality assessment. Data extraction. Extract relevant data from selected studies using standardized forms. Record study characteristics, e.g., study design, sample size, participant demographics, intervention, exposure details, outcomes, and effect estimates, e.g., mean difference, odds ratio. Quality assessment. Evaluate the quality and risk of bias of included studies using appropriate tools, e.g., Cochrane Risk of Bias Tool for randomized trials, Newcastle-Ottawa Scale for observational studies. Consider factors like study design, blinding, allocation concealment, and selective reporting. Statistical analysis and effect size calculation. Effect size calculation. Calculate effect sizes, e.g., standardized mean difference, risk ratio, and their corresponding confidence intervals, c, from individual study data. Ensure consistency in effect size metrics across studies for meaningful comparison. Heterogeneity assessment. Assess statistical heterogeneity across studies using measures such as I-squared statistic and Cochrane's Q-test. Interpret heterogeneity to decide between fixed effects or random effects models for meta-analysis. Meta-analysis. Perform meta-analysis using appropriate statistical methods, e.g., Mantel-Heinzel method for categorical outcomes, inverse variance method for continuous outcomes. Pool effect sizes across studies to derive an overall estimate of effect. Interpretation of results and publication bias assessment. Forest plot. Visualize individual study results and pooled effect estimates using forest plots. Interpret the direction and magnitude of effects, considering statistical significance and clinical relevance. Publication bias. Assess publication bias using methods like funnel plot asymmetry and statistical tests, e.g., Egger's test, Begg's test. Interpret potential bias in study selection and publication of positive findings. Sensitivity analysis and subgroup analysis. Sensitivity analysis. Conduct sensitivity analysis to explore the robustness of meta-analysis results by excluding studies with high risk of bias or outliers. Evaluate the impact of methodological variations on overall effect estimates. Subgroup analysis. Perform subgroup analysis based on predefined variables, e.g., age groups, study settings, to examine sources of heterogeneity and assess consistency of effect across different subgroups. Reporting and dissemination. Prepare manuscript. Prepare a structured manuscript following guidelines such as PRISMA, preferred reporting items for systematic reviews and meta-analyses. Include details on methods, results, discussion of findings, limitations, and implications for practice and research. Peer review and publication. Submit the meta-analysis manuscript to peer-reviewed journals specializing in meta-analysis or medical research. Address reviewer comments and revise manuscript as necessary. Conclusion. Meta-analysis plays a crucial role in synthesizing evidence from multiple studies to inform clinical practice, policymaking, and future research directions in medical science. By following rigorous methodological steps, conducting thorough data analysis, and transparently reporting findings, researchers can contribute valuable insights that enhance understanding of healthcare interventions and outcomes. As meta-analysis methodologies evolve and data availability increases, its application continues to advance evidence-based medicine and foster innovation in healthcare research.